Okay, well, thank you all for coming. Thank you for getting quiet at the right time. Let me welcome Verónica Andreo. Verónica Andreo is a biologist. She holds a PhD in biological sciences and a Master of Science degree in remote sensing for GIS applications. She works as a researcher and a lecturer at the Gulich Institute, which is the Argentinian space agency. And her research is focused on uncovering environmental drivers of vector-borne disease outbreaks. She is interested in the environmental features derived using satellite image analysis, remote sensing, time series, and GIS-based techniques. But in addition to that, she is a part of the GRASS-GIS development team, and currently she serves as the chair of the GRASS-GIS project steering committee. She's a strong advocate for open source geospatial foundation and geospatial free and open source software in general. And she teaches GRASS-GIS courses and workshops regularly. And among other things, she volunteered as a mentor for GRASS-GIS in the Google Code In contest, which is a contest introducing high school students to open source world. And with that, Verónica.
Okay. Good afternoon, everyone. Thank you for joining us this afternoon. Thank you, Vašek, for the introduction as well, and for the invitation. Thank you, Ross, for the invitation. I will talk today about environmental drivers of vector-borne and zoonotic diseases and how we can leverage remote sensing data and products for public health applications. Well, Vašek already introduced me, so the only thing I can say here is that I put a map of where I work. And so if you zoom out, you can see where I work in Argentina. Yeah, there. Basically. So a short overview of the talk. We will first discuss about some motivations for what I do in research, and hopefully it will inspire you to also use remote sensing then. And we will go through some basic concepts about health geography, disease ecology, and then how we use remote sensing for disease ecology. And we will discuss concepts about resolution of remote sensing versus scale of the problems that we study, and how do we use remote sensing for addressing some disease ecology problems. I will give you some examples of works that we have done. And finally, we will reach, of course, the part of the challenges and the gaps that still exist in the use of remote sensing and how we can bridge them. And also some opportunities looking ahead to the future. First, I apologize for my English because sometimes it gets rusty, and especially when I'm nervous, so sorry in advance. 1/6 of the diseases and the disabilities caused in the world are because vector-borne and zoonotic diseases. And especially, they affect people living in the poorest areas and the most marginalized areas of the world. This is why the United Nations consider that good health and well-being as one of the 17 Sustainable Development Goals for 2030. If we look at this goal number 3, SDG number 3, there's a target that wants to end the epidemics and the outbreaks of several diseases by 2030. And among them, we count the neglected, it's not working, the neglected tropical diseases. Why do I mention this? What are these diseases? So these are diseases, usually, most of the cases, transmitted by animals. it's when the animal is might work either as a reservoir or as a vector of a pathogenic agent. So it works kind of, there's an animal reservoir, let's say monkeys for the yellow fever virus. And then there are mosquitoes that bite those monkeys and they transmit the virus among monkeys. But also they are capable of transmitting the virus to humans. And so that's a typical cycle of a vector-borne disease. In some other cases, there are animal reservoirs and direct transmission to human hosts. And those are called zoonotic diseases. For example, the hantavirus, right? It's transmitted from mice towards humans directly. Why do we focus in these kind of diseases? So we can think of mosquitoes, sandflies, ticks, bed bugs, and that kind of bugs. These animals are highly dependent on the environment, right? They are highly dependent on the conditions of the habitat where they live. And what do we do with remote sensing most of the times? We characterize habitats, we characterize environment. We want to learn how the environment changes over time or changes over space. And this is why remote sensing might be such a useful tool to predict this kind of diseases, right? The occurrence of these diseases or the occurrences of their vectors. Now, before we go directly into this, I guess you have seen this map, right? This is the first slide of every GIS course in every GIS course of studies, right? But it was also one of the first times that a map picturing health outcome and environmental conditions was made, right? And this relationship between health outcome and environmental conditions or environment or place, location, is what we now call health geography, right? Within health geography, there are at least 3 fields of study. One is environmental health that deals with, let's call it effects of contamination over human health. So we might think of air quality or air contamination, soil quality, water quality, and their effects over health. Then we have another field that deals with healthcare delivery and healthcare and access to healthcare. So it deals with optimization of how to put hospitals over space and what are the best routes to reach them in case of emergency and that kind of things. And then there's disease ecology, which is the study of infectious diseases and their relationship with the environment. And especially, and by environment, I mean not only like the natural part of the environment, but also the social, political, economical context, because that also influences how we see the environment and what happens in the environment. I will be focusing mostly in uses of remote sensing in the disease ecology field, but we may think of applications of remote sensing for all of these fields, right? And at the end of the talk, or mostly at the end of the talk, I will show you that in our group in Argentina, we are also trying to put a step, put our foot at feet in these other areas too. Now, the main objective of disease ecology, so you know what ecology is, right? It's the relationship of organisms with the environment. So this is basically the same now, but now we are talking about vectors of diseases, reservoirs, or the health outcome in humans and the relationship with the environment. And the main objective is to understand this relationship in order to being able to predict when and where outbreaks of disease could happen. And why are we interested in this? It's because we want to assist decision-making processes and planning of different types of actions to prevent these diseases. So the ultimate goal of disease ecology and health geography is to reduce mortality and morbidity produced by infectious diseases. Now, this relationship can be seen in a very simple way in this kind of triad, the epidemiological triad, in which for disease to happen, we need proper environmental conditions, we need reservoirs and vectors, hosts, and we need a pathogenic agent. Or we can see this in a more complex way with interrelationships and different scales, and one scale affecting the other and so on. But basically what we can see with remote sensing is mostly this part, right? So we can get some information about the weather, we can get information about land cover, land use, how humans use the space, and eventually that will affect the health outcome, humans as well, vegetation, phenology, and so on and so forth. And indeed, the use of remote sensing for health application has been steadily growing. I would say this plot depicts since 2007, but I would say from the last 20 years at least, it has been steadily growing. And the most common remote sensing variables used when dealing with this vector-borne and zoonotic diseases are variables related to temperature, to precipitation, to vegetation, like indexes, like NDVI, EVI, NDWI, land use and land cover data that we can extract from remote sensing analysis, elevation, and so on. In this plot, what we see is the most common variables used for mosquito-borne diseases, right? So LST, so temperature and precipitation, are very important variables there. We know that mosquitoes can survive and reproduce in an area only in certain ranges of temperature and only if they have water to lay their eggs and to develop, right? So this is why they are so dependent on environment. Now, before we choose any of those variables, or the resolution that we need for those variables, we need to consider some basic features of remote sensing data. And these basic features are basically I would say 3 types of resolution. We can speak about 4, but I would say 3 of them: the spatial resolution, the temporal resolution, and the spectral resolution, right? Because this will determine which type of sensor and we will choose, right? Not only according, availability and if they are free or if we have to pay, that's another topic. Let's consider ideal conditions here. So if we want, for example, to study weather and we want to see storms forming and evolving, we need something with a very high temporal resolution, like in terms of minutes. Every 15 minutes, every 30 minutes max, we need an image. And we need a large extent, right? So we can, we can prescind of very high spatial resolution because we really don't need it, but we need temporal resolution. But if we want to study, for example, urban growth, then we need a higher spatial resolution, like 10 meters, 30 meters, something like that. And we can study that over years. So if we have like 1 image per year, aggregated data for every year, that would be fine, right? Because we want to see the spread. So it basically, which data we choose basically depends on this relationship. And then, for example, we want to see diseases in plants. Well, we will need a sensor that has bands that allows us to see in the near infrared part of the spectrum, right? Because that is specific for plants and for plants' health, right? So all those features we need to consider. And we also need to consider how the variables that we will be using affect or at which scale they operate as regarding to the phenomena that we are studying. So let's say we study species distribution, right? Mosquito distribution or a tree, how it is distributed. If we want to study how it is distributed all over the world, we will use climate variables because climate operates at global to regional scales. If we want to know if in this room or in the next block, the tree will be or not, well, that will depend on more local variables. So then we will try to observe biotic interactions, if the competitor is there or not, how's the elevation, yeah, something kind of zooming in, let's say, zooming in a little bit, and there will be other variables involved. And the same will happen, the same kind of relationships we will observe according to different taxonomic groups. So if it is invertebrates or mammals or what they eat, let's say, or in different aspects of plant physiology or ecology. So if we want to study the leaves, we need high resolution and high temporal resolution. But if we want to study a succession in ecology, then we need like longer time series data, right? And the same for animals. Which type of movement, if it is daily movement or if it is migration, again, the same. These plots are really interesting. I leave you the citation, the references there, because these plots are really interesting to understand which satellites have been used already for these kind of studies. Now, all this said, how do we use remote sensing for disease ecology? We use this pretty common workflow in which you have a response variable. Basically, in this case, it can be human cases of a certain disease or counts of vector animals or reservoir animals, and so on, incidences, infections rate, right? We have predictor variables. Then basically we fed that into a model that can be something super simple as a linear regression or something much more complex like Bayesian statistics, deep learning, machine learning, and so on. And we get predictions in different spaces or in different times and spaces. And we can use remote sensing either for mapping the response variable or getting the predictor variables or for validating the predictions that we make. Now let's see some real cases, some real studies about these examples. For detecting and mapping species occurrences, for example, we will need, of course, very high resolution imagery. We won't see a mosquito with the satellite, that is so yet, let's say yet. Okay, but we can see, yes, colonies of penguins, for example, if animals are gregarious, so we can detect groups. We can already count elephants or cows, right? If we think on a more global definition of health, we are also interested in animals' health, right? And we can also do like this example here for great gerbil burrows. They produce such a modification of the habitat that you can get indirect estimations of their abundances. So when they build these burrows in these kind of desertic habitats, you can basically see them from satellites. And you can then classify if the burrows are occupied or not, if they are new, if they are old, and so on. So I find this really interesting. Of course, it can also work for plants, no? And you can also detect infected plants like this pine beetle infection with satellite data. And then you could model how the pine beetle infection is distributed. No? Or you can also use remote sensing for the predictor part of the workflow. And in this case, for example, we reconstructed the whole MODIS LST archive. So it's 4 images per day, the 4 passes of MODIS. And we did temporal and spatial reconstruction. And once we had the whole data like gap-free, let's say, we estimated different relevant indexes according to mosquito ecology. So we knew the optimum temperatures in which they live are, I don't know, 25 to 35, let's say. So we counted how many days per year we have such conditions, or how many days per year we have temperatures below -5 degrees, which is like the minimum limit for survival, and so on. And then all those indexes, we applied a co-clustering analysis, which kinds of detect clusters or groups of both the temporal dimension and the spatial dimension together. And so you get groups of years and groups of pixels per year that behave in a certain way. And then basically we use the West Nile virus cases over those clusters to detect when and where those conditions were met, let's say. And the reconstruction part and the estimation of indexes was, of course, done with grass. Yes. A different use for MODIS LST data is to estimate this environmental risk of dengue that we do in the Argentinian Space Agency. And in this case, again, it's quite simple. The algorithm is quite simple because you use a threshold of growing degree days that the virus need to replicate within the mosquito stomach, basically. So this is also temperature dependent. And according to temperature is how long does it take for the virus to replicate within the mosquito, and then to be able to infect the next person that the mosquito bites. Something like that, I hope to be clear. And then according to the number of potential cycles that the virus could do, we characterize the risk. So the higher the number, the higher the risk.
What are the points on the map?
The points are different localities in Argentina, so cities basically, higher than, yes, sorry, higher than 5,000 people.
And the red areas are
the highest there is, yes, okay. Yes, actually. And you see where I live is one of the, it's usually one of the hotspots, yes. Then we used, this was the first use case where I started learning grass. So that's why I have like a special love for this example. We use species distribution modeling and GIS-based approach for hantavirus pulmonary syndrome risk map. So basically we follow the workflow that I was showing before, but not only for the mice, but also for the human cases. And so we ended up basically with 2 maps, one for the mice, for the reservoir, and one for the probability of human cases, probability of transmission of the disease. And what we did was just basic GIS. We rescaled those maps, and then we put them together. So the reddish area here are the areas where both the mice and the human cases are most likely to occur. This disease, so I did my PhD with this disease. And the problem with this one is that not only you can get it from a mouse, but you can also get it from other humans. So it's one of the only ones that can also be transmitted human to human and has a mortality rate of 50%. So yeah, the first time I made a mistake while removing all the equipment, I was really scared, really scared. So this was almost, you see, like large scale, let's say, or large extent, but sometimes we have more localized data, no? And we start getting creative in generating variables, in generating predictors. And these dots were cases of cutaneous leishmaniasis. It's a disease that produces some wounds in your skin. And it's transmitted by a sandfly, which is similar to a mosquito, but different, basically. A little more blunt, let's say. And this sandfly has the particularity that whenever there's environmental change, like think of urbanization or deforestation, this species is the first one that comes in and colonizes and increases their abundances immensely. So the people working there then have more probability to get the disease than anywhere else, right? So knowing this aspect of the ecology of the sandfly, what I did was, okay, let's see if before the outbreak, there were environmental changes that we can detect from remote sensing, so we can associate that to the occurrence of the disease. And basically, I applied something super simple, but a change detection algorithm, which is change vector analysis. There's the module link. It's an add-on in GRASS. And once we detected the changes that you can see here, so here was an urbanization, here is flooding, and so on. We estimated the distances to those changes, and we put them in this workflow of species distribution modeling. And these were the most important variables in the modeling. So the distance to environmental changes explained most of the distribution of cases. Again, GRASS. This other example of using remote sensing is a bit more tricky, but I really enjoy doing this, carrying out this study. It's with mosquitoes, not with mosquitoes, with eggs of mosquitoes. So in the city of Cordoba, there's a network of ovitraps. Ovitraps are, let me show you this. So it's basically a black container with water and a tongue depressor that you use to monitor mosquito activity. Why? Because the females lay their eggs in this wooden stick, right? Like in the level of water. So water is here, mosquito put the egg here. And then when water rises, it gets the eggs and then the eggs develop, right? So they have that. And they have weekly monitoring of these ovitraps over 3 years, more or less. And so I thought, looking at the whole time series, you see that there are different shapes of the time series. And I thought, okay, perhaps there are different patterns in the activity of the mosquitoes, and that could be related to risk. And if that is also related to environmental conditions, we can predict all over the city where we don't have ovitraps, which temporal pattern is that reflecting? So basically, I collected all the time series. I did time clustering. It's the special kind of clustering with temporal distances, so dynamic time warping and that kind of things. And we found 3 patterns. And they were differing in the length of the mosquito activity, in the peaks that they reach, and so on. And so then we use Sentinel-2 data of the, let's say, of the area surrounding that ovitrap, and we applied machine learning to predict all over the city of Cordoba. The problem was that the dengue outbreak happened during the pandemics, and all the samplings were stopped by then. So we couldn't actually do the relationship between the temporal pattern and the dengue outbreak because they were like sequential in time and not simultaneous. But well, it was really fun work to carry out. And as you see here, well, it's really small, but there are the ovitraps. We were not happy with the distribution of the ovitraps because they were like in 5 groups. And that does not reflect the environmental variability of the city. So we don't know what environments are we seeing here, no? And so with the master student, we applied, again with grass, of course, we applied this object-based image analysis to classify a very high resolution image of Cordoba. And then we estimated landscape metrics for the census tract polygons. Cluster, we did a k-means clustering of those polygons, and we found 4 different groups, like representing environmental conditions. And then we distributed again the ovitraps, but in a stratified way. So this can be useful not only for ovitraps, but for any other kind of sensor that you wanted to reflect environmental variability. And we use grass and R, basically. This is one of the last ones, I promise. Here we used remote sensing. So we use remote sensing for a lot of spatial prediction. But here we use remote sensing mostly for temporal prediction. And basically what this system does is it downloads temperature and rainfall data from NASA and NOAA for a pair of coordinates, in this case, my city. And it uses it to run a population dynamics model of the Aedes aegypti mosquito, and then predicts the abundances of the 4 different life stages of the mosquito. So eggs, number of eggs, number of larvae, pupae, and adults. The interesting thing is that you can not only use current or historical temperature and rainfall, but you can also use forecasts. So if you run the model with, let's say you run it today with a 15-day weather forecast, you can know in advance a prediction of what would happen with the mosquito population in the city. And now there's a PhD student also working with us that will try to spatialize this model, but to make it predict, let's say, per census tract or per city block within the city. Not only like for the whole city, as in a mean, let's say, but within the city. And for that, we will be using higher resolution remote sensing and try to put those variables within the model. So let's see what happens with that. And last, we are now developing with different other institutions like NGOs and companies and so on. We are developing an online surveillance system for vector-borne diseases, which basically covers the whole workflow from download of satellite geospatial and satellite data import and process into GraphGIS to obtain the variables. Then basically R reads all that data, performs the modeling, and we export the results of the models and publish them in a WebGIS. We use Zoonotic for that. So it's not yet public, but it looks more or less like this. And you can make your maps, heat maps, and you get charts and counts and so on. So these are like dengue cases in 2020, for example. And there are, I won't go through this, there are different other projects from PhD students in the lab, and some of them are using, as I said before, remote sensing air quality to predict asthma, for example, or respiratory diseases. So we go into the environmental health part, and there's another student that will be studying healthcare access of native populations in northern Argentina. So it's very, very marginal conditions there, very poor conditions. And we want to understand how they access, if they access to healthcare, and how to optimize the distribution of resources there. So once we saw all these examples, is everything studied already? No, not at all. There's still a lot of things to do, a lot of space for improvement, a lot of space for new techniques, new data that is coming, new missions that are planned. So there's still a lot of things to do, and there are still challenges and gaps that perhaps we will have to deal with some of them for a long time. Perhaps they are just inherent to our profession, let's say, and some others can be breached. So this trade-off between the different remote sensing resolutions and the problem that we study, well, that's something that we will have to deal with, like every time we start a new project. That is the part that perhaps artificial intelligence cannot do. Then if you work with optical data, so visible and infrared, you will always have the clouds, right? That will be affecting the temporal resolution that you want to get, right? So you either go to spatial and temporal interpolations and reconstruct the data, which has their errors, of course, or you just aggregate data and sacrifice, let's say, temporal resolution. There's also need for performing a lot of corrections to the data. So spatial agencies usually provide different levels of processing. And if you are not happy with the highest level of processing, you need to do the corrections yourself. So that means geometric correction, radiometric correction, topography, atmospheric, and so on. And in these last two items, I'm speaking from the global south, basically. We still have limited access to very high resolution data, to LiDAR data, to hyperspectral data. It is costly, basically, for us. And also in general, for example, hyperspectral data especially is pretty tricky to scale yet. It's not really global data because image size is so large that satellites cannot be taking images all the time as Sentinel or Landsat does. So it's like by order, let's say. So a user programs an image, and then when the satellite passes, it takes the image, basically. No? And there are upcoming missions to try to solve that, so we will reach there. And then again, from where I come, there's still need for a lot more of investment and capacity building, because we are dealing with this increasing amounts of data available, but at the same time, we still have very limited bandwidth, storage, and computational capacities. And yes, there is the cloud and cloud computing and parallelization and so on, but that also requires resources. So yeah, it is an unfair world, let's say. But I think a lot can be done for this. And last but not least, field data will always be needed. So we cannot say anything if we don't either train or validate with field data. Whatever we say without it might be a complete nonsense. So we must calibrate whatever we analyze with remote sensing with field data, right? And that is also something that takes time and resources. Now, there are, I think, some low-hanging fruits, or more or less low-hanging fruits, to cover, to bridge some of the gaps. And for example, if you are bothered by clouds, you can use radar data. There is radar data for free. No? So you can use SAR data. And for example, there's this newish satellite from the Argentinian Space Agency called SAOCOM, which has an L band. And that allows to estimate soil moisture, for example. Soil moisture is pretty relevant for a lot of insects that lay their eggs and start their development in the soil. So for, at least for my field of applications, that would be one good way to go. There's also now some open LiDAR data in the JEDI on board of the International Space Station that was renewed, I guess, for one more year or something. So there's already 1 year of data, and I think it was renewed for one more, and it covers most of the world at the resolution of Landsat satellite, more or less, 30 meters. And there are some open source, some promising open source solutions for the cloud, like OpenEO Cloud, Actinia, and perhaps Open Plains. I hope to see Corey here. But, so I think there are some stuff that could cover those gaps. And as I was saying before, there are a lot of new hyperspectral missions. Some are already in the space and some are approved. And indeed, Prisma, the hyperspectral Italian satellite, had opened their archive like last week or something like that. So now you register and you can get access for free to the archives. Before it was only with agreement, and that takes forever. And finally, there are now a lot of specialized cameras on board of drones. So drones have gotten a lot cheaper, a lot more accessible, and cameras as well. And there are, for example, hyperspectral or multi or hyperspectral thermal cameras that would allow this sort of application. So that is counting monkeys from a drone with a hyperspectral thermal camera, just because the guys regulate temperature, so you can detect them. No? And I found this like, wow, I'm so nerd that I love this. Yeah, I was even telling my nephew about this, and he was like, so I think there is kind of a bright future in terms of technology, only if it was accessible to everyone. But there's a lot to do. So in conclusion, I think there's a lot of space and a lot of things to do with remote sensing. There's a lot of potential applications and developments to do. And remote sensing has a big deal to contribute to this view called One Health, that is humans can only worry about their health if the environment is healthy as well, and if the animals are healthy as well. And we have like a clear proof with COVID, no? So it's really important that we not only worry about our health, like killing mosquito, exterminating mosquitoes. That does not exist. We have to have a wider vision for that. And remote sensing precisely gives that this wider vision, this systematic vision. So thank you very much. I hope I didn't go so long. I want to, of course, thank my colleagues at the unhealthy lab. We are all but healthy there. Yeah, I don't know. Science is a team effort as well. I need to thank them. And then all the references that are cited throughout the presentation are here. So then I can leave you the link and you can access them if you're interested. So yeah. I'm open to questions if you have.
Yeah, thank you. We have time for questions, and we're going to be using mic. Vero will try to repeat a question for the online audience.
Was it working in the end? Or?
Well, this one hopefully, yes, not the.
Okay.
Okay, so question.
Yes.
Yeah, you mentioned a lot the resolution and scale and issues like that. I was wondering, how do you deal with when you need to, I assume that you are, in your studies, you are combining data from different sensors. Like if you are combining data from MODIS and data from Landsat.
That's tricky.
So how do you deal with the differences in resolution, and how important essentially is, can you just resample like with nearest neighbor, or do you need some more sophisticated resampling, or you just end up with the lowest resolution?
Yeah, I would say the politically correct answer would be you should go to the lowest resolution. No. In the use cases, sometimes we just don't, and we go the other way around. But I think the question is, do you really need to combine such different resolutions? Like, which is the scale of the problem you are trying to answer? No? So if it is at the landscape scale, why would you want to use something of a 1 kilometer resolution like temperature? Will that really affect, or you can solve that using elevation at 30 meters and then you have the same? No? So it's.
So how do you get the temperature, for example, if you are doing just 30 meters?
Well, you would have Landsat there, but then that is not, so Landsat has thermal sensors as well. And I think they are at 60 meters originally, but they deliver at 30. That is not so critical, I would say, comparing to 1 kilometer to 30 meters. So you can do nearest neighbor there. It's not so big deal. But again, what do you want to predict with temperature at that scale? No? If you're trying to predict mosquitoes, well, temperature at that scale, I don't know if it is really relevant, unless you go even more microclimate and then use sensors. So it's really a compromise there. Yeah.
For your hantavirus work, did you have an army of mammalogists out there documenting where the rodent populations were? And what came first? Did you identify a case and go in and do the rodent work to know where the rodents were?
Sorry.
For the hantavirus work, we had.
Yes.
You actually.
For the hantavirus. Yeah, for the hantavirus work. So I did my PhD in southern Argentina. We did samplings there. So that was kind of a landscape scale because it was like kind of a small area. And for the other, for the one that I presented there, it was moreover literature review for records of all the mammalogists in Argentina catching mice, let's say. So I got that data and related that with mosquitoes. And for the human cases, I got them from the health ministry. Actually, from a person that I knew at the health ministry, because it's really tricky, really tricky. Yeah.
Have you tried to overcome the lack of LiDAR by using drones and using optical open drone maps to do structure to motion?
We are getting there. So I haven't used LiDAR myself, but they are getting a drone in my institute in Gulich. And so we are all like eagerly waiting for it to start doing research with that as well. I haven't used LiDAR myself.
I was just looking at that. You were talking about microclimates for urban areas.
That would be really cool.
If you had the elevations of the buildings, could you be considering something like in the solar radiation model
Yes,
to determine where the, okay.
yes. Indeed, we wrote a grant to get a lot of equipment, but it was rejected. But we wanted to do that kind of things within the city with the LiDAR scanning. So to get like potential breeding sites for mosquitoes and identify that and different heights and so on. But we didn't get it. So, yeah. And then there, would you deal with data privacy problems if you want to fly over the city? So there was oh, sorry. Sorry.
No, it's okay. First, I want to say thank you. It's a very wonderful presentation. I was wondering, you mentioned that on the ground data is always going to be needed. So I was wondering, is it common or popular in Argentina to incorporate citizen science data or like volunteer generated data into these types of analyses, or is it kind of, I guess, lacking of the amount of data?
It is in, how you say, in diapers. So we are starting. Even the health ministry created something like really simple, like a Telegram bot. So people would send pictures of breeding sites for mosquitoes or pictures of mosquitoes, and then they would get like, okay, this is the species or things like that. And one of my students, the one working with air pollution and asthma incidents, he will be also working with yeah, let's call it citizen science, doing like working with students that visit CONICET, that visit the spatial agency to get some data about asthma incidents and where they live and when do the people get their kind of acute episodes of asthma, and then try to relate to what happened before in terms of pollution or changes in the land use or fire, things like that. But it's just starting. It's just starting, yes.
I was especially wondering with like the dengue study, just thinking about here with COVID, there was a lot of voluntary reporting, and so I was just wondering if y'all had something similar.
Yes. Yeah, that would be really great. It's something that in the institute is one of the lines that we are like willing to address in the near future. But as I was telling Vasek, we are kind of a small institute yet. So yeah, we need more people there and resources to develop more things. But it's really interesting. It's also really noisy, that data. It can be really noisy. So you have to do some filtering as well. Do you work with the, yeah. Yes. But the nice thing is when you are used to, I don't know, a sample of 30 10, 20, like in ecology. And then you see, I don't know, tens of thousands of data. It's like, oh my God. But then it's pretty noisy as well. So you have to clean that. Yeah.
I did have one more question.
Yes.
if That's okay. Earlier in your presentation, you mentioned that by 2030, there are all these goals to eliminate.
Yes.
You, with the challenges you mentioned with remote sensing, like you think that that's an achievable goal?
No, not at all. I'm not politically correct as you might. No, I don't think that's achievable. It's 2023 and we are having a terrible dengue epidemics, for example. So I don't think it's something achievable. There is quite some, I mean, people are getting worried, let's say, but clear actions, I don't think they are being taken yet. Yeah. Yes.
How do you balance the trade-offs between presenting your hotspot maps to the public when you put it out publicly and creating like an overestimate of fear of these different diseases?
Oh, with a lot of care. We haven't yet published this this WebGIS, no? But in my experience has been with like interviews, for example, or things like that. And yeah, you have to be like super careful. And people from the health ministry usually kind of smooth what we say in our results, for example. And there's also like, you want to be faithful to the truth, let's say, to what you found, but you also want to keep the collaboration with the health ministry. You don't want to like put a bomb there and make it explode. So yeah, we are kind of training ourselves to be careful with what we say. And then sometimes we directly pass like maps to them, and they are the ones deciding what to publish or not, or if they use it only internally, like the minister or things like that, and they are the ones explaining. We also did, it's nothing to do with remote sensing, but we did an application to count mosquito eggs in ovitrapes. So it's online, you take a picture of the ovitrap, and then you, with the slider, you count how many eggs are there, and then you get plots and so on. And it was really tricky to, this collaboration was really tricky because we went with them to the field. We were first thinking of a mobile application, but when we went with them to the field, what they wanted to do is just finish quickly. So we were with the phone, taking the picture, the phone was crashing and so on. The phone won't work. No way. But it's really interesting collaboration. So yeah, if you handle it careful, with care, it can work. Yeah, Owen?
For some of these online portals that you have for real-time updating and monitoring, do you guys make uncertainty information for some of your predictions, or is that kind of-
In this online platform, what we do is we run two different models. So we run Maxent and random forest models, and then we do ensemble modeling according to weighted by AUC. And we estimate also the standard deviation between the two models. That's how far we reach, we get with uncertainty. Yeah.
No questions online.
Yeah,
It was all clear.
right on time. Let's thank Veronica again.
Yeah.
Veronica will be here actually the next week. So if you want to meet her, you can tell me or feel free to email me, and I can set up a meeting. And tomorrow, in the morning, starting 9 a.m., Veronica is leading a studio with the title. Do you remember it from top of your head?
Remote sensing for species distribution modeling with grass and R. Yeah, basically, what we do in this online surveillance system, we will basically follow a workflow similar to that, using grass and then calling grass maps from R and doing the modeling, writing them back to grass. It's nice.
Thank you.
Thanks again.
Tomorrow.
Oh, the chair.